Hello again to our dynamic duo here, Georgina and Joan. We're going to abstracts and the winners of the contest for the best. Yeah. So I'm going to get out of the way.
Hi, David. Hi, everybody. Okay. I like the photo of Maximilian. Okay, so we start with the first. How did we say we're going to start with the third going to the first? Okay, so the third award goes to Maximilian Anfried from the University of Singapore. Max.
I'm here.
Hi.
Hello.
You're gonna be talking today about lipid clocks.
Correct. I'm just sharing my screen and hope you can see it.
Yes.
Perfect. So, good evening or good afternoon to everyone. And thank you for targeting Metabesity to let me give a talk and give me like an award. That was like great. It always feels good as a young scientist to get like some feedback from like just people and the community in general. So I'm gonna talk about lipid clocks. And obviously the first question is, do we really need like another clock, right? We already have like our epigenetic clocks, we have transcriptomic clocks, you name it. But as Nir actually just said on the last panel, we will have like a lot of biomarkers, and they can be used like for different things, like normal clocks that we had like throughout the centuries. Certain clocks are better at measuring like various things, right? I mean, while the sun is shining and you don't have electricity anymore, maybe like a sun clock might actually be like more valuable than like a digital clock. And I think it's like similar with like all those aging clocks that we currently build on different kind of data, that we will find different uses for them, that one might be better at predicting like a certain disease outcome or like an age acceleration than another one. And why lipids? I mean, we know that lipid composition changes with age. We also know that lipids can be modulated through diet and drugs. And lipids are usually part of like a normal a panel in the clinic, which is nice compared like to methylation, where I think it might be like a little bit tougher like to get it like really approved into hospitals. So for like the people out there who are like not that deep into lipids, here like just a quick summary what lipids are. I think what strikes me always crazy is that we actually don't know how many lipids are out there compared like that we know how many proteins are actually out there. But lipids are like this diverse and heterogeneous set of small molecules. They're usually classified into 8 categories. We have more than 80 main classes, 300 subclasses. And I think like just 2 months ago, they added like new subclasses. So it's like growing. We find more lipids or like classify them better as we do more research. And obviously like a lot of lipids are like we use in our like daily vocabulary as like biomedical scientists and doctors, like cholesterol, testosterone, estrogen. They're like relevant for aging, but also like sphingomyelins or like THC, like from the cannabis plant. So lipids are all around us. They're like mostly hydrophobic, and we have variation between species, but we also have like conservation. And if you look like for lipids and the role in aging, we know that like signaling lipids, I think, are like the most interesting for modulating aging. It's like especially like those steroid hormones like estrogen and testosterone, but also like diacylglycerols and prostaglandins and the like. And then we have like structural like lipids, more like membrane lipids, which I always think are like might be more robust like for measuring aging, like glycerophospholipids or like ceramides, cardiolipids and the like. And then there's obviously the entire category of like energy storage, where like the most prominent are like fatty acids. So what I was thinking is, can we use the lipidome to like estimate biological age? And we started like with a proof of principle in worms, where we had like normal aging worms, slow aging worms, and worms with accelerated aging, longitudinal over like different time points from day 3 to day 20. And we collected 168 lipid species like across like the lipidverse. And first, if you look at the principal components, you can actually see that like at day 3, all the lipids of all the different worm strains are actually kind of clustering together. So they have like similar initial lipidomes to start with when they're born. But then as time passes, they are actually diverging. And like the day 10 wild type lipids are like together clusters with like the slow aging ones at day 15 and 20, which kind of makes sense, So the slow aging ones at day 20 are like more similar to the wild type at day 10. What is also like common across aging clocks, that usually the first principal components correlate well with age. We see that here as well, like with principal component 2 is like correlated with age for all of them. Principal 1 and 3 a little bit less, but still a strong signal. And then we can like train this lipid clock with like a simple elastic net regression. And what we do, we train it on only the wild type data and the very young mutants. And the idea behind is that if we put in the mutants at the beginning, maybe like certain lipid changes that are actually really depending on the mutant and not on the age are already incorporated into the lipid clocks. If we then train this clock, we can see that the black line here is like the wild type data. And obviously that was in the training set, so it's a perfect fit. But then the red one is all the data that was like not in the training set, and those are the fast aging ones. And we can see they're like aging way faster, roughly double the with double the time than the wild type ones, whereas the green and the blue ones age with only half the aging rate of the wild type ones, and those are the slow aging ones. So it also like makes sense that we in a way can use the lipidome to predict like aging trajectories or like the rate. We also use that the rate then and some Monte Carlo simulations to actually simulate potential trajectories. And you can see it here on the right, where the very thin lines are the simulations, and the thick lines that are zigzags are actually like data from real experiments, and there's like fairly a good overlap. And we published that like in May of this year. But obviously, like worms are like well, we can also like make an interpretation of like this data and see like which lipids are actually changing the most. And what we usually get is like a lot of LPCs and ceramides are changing. And the question is obviously worms is one thing, but does the same like work in mammals and eventually humans. So we built like a brain tissue clock for like the mice of mice, for the brain of mice. With like we took this data from this Metabolome atlas that got published end of 2021, which has like 64 mice at different time points, 400 samples in total, because like 10 brain regions per mouse, and roughly 820 lipid species. What is interesting, what we actually find, that like that brain regions that have neurogenesis, like the hippocampus and the olfactory bulb, have a predicted like lower lipid age than like, for example, the cerebral cortex. And we also see like that the hypothalamus and the olfactory bulb for like different sexes also ages differently. That like for the hypothalamus, like that the male ones age faster and the female ones age slower. And for the olfactory bulb, it's actually the other way around. Furthermore, we like looked into, well, human data, right? Because that's what we eventually want to get to. Can we use lipid clocks like to measure it in humans in the clinic? And that's like another lipidomic data set that we used like from the internet, and we built like a clock on like 70 healthy people with like a normal BMI, and then we evaluated it on overweight and obese patients. You can see like on the left, it's a fairly linear function again, which is like fairly similar as for like a lot of clocks, that like between the age of like 20 and 80, it's like fairly linear, and then we have like more like sigmoid or non-linearities in there. But what we also find is like that the lipid age is actually accelerated like for overweight people by roughly 4 years, and for obese people by roughly 6.6 years. And I think Neil mentioned earlier something similar, that usually like biological age like for obese people is like also higher. So and methylation clocks stay the same, and we see the same like in lipid clocks. So that advantages and disadvantages to those lipid clocks. Lipid clocks are like already part of, or like lipids are already part of the clinical biomarkers, which makes it potentially convenient to just say, can we measure 20 more lipids in the clinic and then use those for a clock? We also have like a direct connection to diseases in the biology. They can be quickly manipulated to diets and pharmacological interventions. So we don't have like to wait those 3 months, because they react often way faster, like within like a few weeks. And also like all species have lipids, whereas like not all species actually have like methylation. So it might also be like just good like for like a lot of those preclinical tests like worms for like measuring the age of a worm, or the biological age of a worm. Obviously, the disadvantages, I always think of it like lipidomics is like where like proteomics, it was like 15, 20 years ago. There's like not a lot of standardization yet on those panels. We still don't know like which lipids are like really relevant for those clocks. But obviously that's like great like for like young researchers to explore it. And there are like also like not that many large scale data sets yet that we have, like, for example, like for methylation or transcriptomics. So in summary, we can use the lipidome to estimate biological age of tissues and organisms. Some of the results are like matching like with epigenetic clocks. What we found in like from worms to mice to humans at ceramides usually have like a lot of strong weights in those clocks. Hopefully they can be accepted faster in the clinic. And with that, I want to acknowledge like the people that supervise me and help me out, like Brian Kennedy, my main supervisor, and Marcus Wenck at University of Singapore, then Jan Gruber at Yale NUS and his team, and Morten Skybæk-Knudsen in Copenhagen. And with that, I come to an end. If you have any lipid data, feel free to reach out to me. And yeah, I'm happy to answer any question.
So David, do you want to start with a question?
Maximilian, great job.
Yeah, sorry. Yes, sorry. Congratulations.
Thank you.
Yeah, hi, Maximilian.
Hi, David.
So there's a couple of questions that come into my mind and criticisms as well. So overall, biological clocks are being used in multiple ways and will be used more and more in the clinic as we evaluate anti-aging compounds and interventions in general. So this lipid clock definitely adds to that, right? And I'm very supportive of this, and that's why we chose your abstract. That's one of them, the winners. So the question that comes into my mind is, did you have so many different things? So for example, did you were you able to validate or to evaluate how those lipids in your worm data change in other cases? There's data on mutants that have shown, for example, accelerated aging in different settings, and that's well known and well established, especially at the Buck Institute. A lot of people are working on worms and have a number of mutants. It could be tau, beta amyloid, like overexpressing. It could be other types of mutants that show accelerated aging. And so all those data are available. It would be very important for you to see, look to see those your lipid clock shows differences in these cases. And in addition to that, there's been a number of different interventions, right? So we know the Buck at least have the pharmacopoeia of the anti-aging compounds, and many of these things have been published. So on the one hand, you have the accelerated model for aging that you can test your clock and see if that's actually increased. And on the other hand, we have interventions that are well established that you could look to see if that's decelerated, right? So those are the first set of questions, and then I'll have a few more.
Yeah, sure. I mean, absolutely. And that's what you just mentioned is currently in the works. We're currently like looking like to collect more data, or like you said, there's already data out there to see if like the worm clock really matches. What we to some extent found is like what was like nice about this data that we had in the lab is like that all the data got like collected in the same lab by the same people at around the same time, which in a way obviously like limits its batch effects, right? So I think it was like just like for like a proof of principle, that's what you want to have in a way, that you can see, does it actually work, or is your theory correct? But you're right. Ideally, you at the end, what comes out of it is like a lipid clock that can be used by every lab that collects like lipid data of worms and tests interventions and get like a clear signal on there, because like there's like no like, because it's like independent of like different labs and batch effects. And then yeah, evaluating that with like interventions like rapamycin or whatever you want to test in worms, or like new molecules that we'll find, is obviously like probably would be like the gold standard for a worm lipid clock in that case.
Yeah, that's helpful. One more thing, if I may, Georgina. Yes.
Of course. Yes, of
Okay.
course.
So then you go on and analyze mouse data, and then finally you look into human data. My concern is the mapping of those metabolites or lipids from one species to the other. And my guess is those are not necessarily the same, right? You're selecting features that may or may not be that may not overlap between species. And my guess is they don't. Did I understand that correctly, or did you?
So your question was if like the different lipid species are like translatable like to from one organism to the other?
Right.
So to be fully
when you did your human analysis, you retrained the model entirely. You didn't use the selected features from worms, did you?
correct. It's like an entirely new data set, like different kind of species on there. And here's like one thing is like, in a way, lipidomics currently making like a lot of progress, right? So like a data set like from 3 years ago, 4 or 5 years, at that time, they mainly collected 300-400 different lipid species. Whereas like nowadays in 2022, they're like more than 1,000, 1,500, depending on laboratory, right? So people adding to that. And that's obviously great because like you get like more lipids in there, but at the same time, it might make not people or like data sets as compatible as they would be, right? I mean, for methylation, you always have like your Illumina kit with like, it starts with 27,000, then the 450,000, now you have the 850,000 epic arrays, right? And lipidomics currently doesn't have the standardization, and there's like no real panel where, and often like the panels that they use for lipidomics is like also depending on the study. If like a certain study want to look more at the brain, they might select different lipids that are interested in because they have like always like a prior of what they think what is important. Whereas so that there is, to be fair, there's like some bias in how people collect the lipid data on what they want to see or what they expect to see, which makes it obviously tricky to build like a generalizable clock.
Question, do you know how diet modifies the lipid clock and high-fat diet in particular in humans?
Not yet. I'm trying to get my hands on a data set like that, and I'm working on it. But that would be very interesting to see that if you have like a lipid clock on like the serum and then like different diets. I spoke to a few people that said they have lipid data on that, and I hope I get it soon to test it out.
That just may be a tricky part, depending on what people ate that day or that night. It may change their lipid age.
Yeah, I wonder if
it's
you really would see that, if you would really get like this very short changes, or if you see like more like a mode in there, or like if the lipid clock actually selects certain lipids that actually are independent of nutrition, and some lipids that might be depending on nutrition, or like exercise and other things. I currently don't know that.
Yeah. And then one last comment, if I may. It's about translationability and the possibility of making this a diagnostic test, right? So going all the way to the clinic, and one of the incentives or promises that you mentioned at the very beginning is that these things are readily available in the clinic, and they're not. Right? I'm pretty sure that most of those species that you have selected in your panel are not commonly measured in the clinic. So it'll be somewhat of a tricky step up to try and have this implemented in a CLIA setting for a diagnostic test. Most of these species are quite rarely measured, and I don't think they're actually available if you don't go for mass spec.
Yes, but here's the thing that I think about is like, and maybe I'm in a lipid bubble there, but a lot of people start talking more about ceramides and all the relationship to diseases, right? And I could see that maybe hopefully in the near future, right? That maybe like there might be like a standard ceramide panel that might be used more often, like measuring cholesterol. Some people talk maybe you should measure ceramide similar like cholesterol for like certain diseases, and that would be super helpful. But yeah, you're right. Currently, you need mass spec. It's not that trivial.
Yeah, thanks.
So I think we're going to move to our second prize winner, which is Mohammed Khan, who is going to talk about his studies looking at deregulated mTORC1 signaling and mRNA translation in Parkinson's disease pathogenesis.
So thanks, M2022 for this opportunity. So my name is Mohammed Khan. I am a research associate with Dr. Valina Dawson and Ted Dawson and Johns Hopkins School of Medicine. So today I talk about deregulation of mTOR signaling and mRNA translation in Parkinson's disease pathogenesis. Deregulation of mTOR translation is quite evident in familial Parkinson's disease. As you may know, the best example is dominant LARC2 mutation in familial Parkinson's disease. The mutation in this kinase leads to abnormal protein synthesis and dopamine neuron loss. Direct evidence in another kind of familial Parkinson's disease, for example, Parkin and PINK1 recessive mutation in the genetic background, there is no direct evidence of translation regulation. However, this paper, they show that the overexpression of translation, activation of translation inhibitor 4EBP in this PINK1 and Parkin genetic background, it rescue PD pathology. There is a crosstalk between mRNA translation factor and alpha-synuclein toxicity in alpha-synucleinopathy. You may know the findings from Dr. Susan Lanquist's lab from MIT. She showed and the overexpression of mRNA translation factor, it detoxifies alpha-synuclein toxicity in this model. Finally, this study in a mouse model, they show that the overexpression of translation inhibitor 4EBP, it protects neuron from alpha-synuclein misfolded alpha-synuclein toxic stress. And this is really, really interesting because all these data, it say the translation pathway, both in familial Parkinson's disease and sporadic alpha-synucleinopathy may crosstalk with the mRNA translation pathway. So we came up with the hypothesis. The hypothesis we said that as alpha-synuclein interact with translation pathway. However, there are a lot of questions with this hypothesis. The most important question here, as you can see, whether are there anything that pathological alpha-synuclein is interact with mRNA translation. This is one of the questions we struggled in a couple of years, and we figured out actually maybe a pathological alpha-synuclein directly affect mRNA translation. So the approach we take is called complementary proteomic approach. We wanted to see whether pathologic alpha-synuclein or aggregated alpha-synuclein it have any connection with endogenous protein complexes. So we take the proteomic approaches, and I will explain in the next slide. So the models we use almost everything. We use neuron, we use Drosophila, we use mouse model to establish our hypothesis. So as I mentioned in previous slide, we use the complementary proteomic approaches to find, to show pathological alpha-synuclein interact with both mTOR pathway and translation pathway. So in first approach, so in Hexel, we express the aggregation-prone A53 alpha-synuclein. It fused with the enzyme is called ascorbate peroxidase, and we do the in situ labeling to see what kind of endogenous protein level by alpha-synuclein. In second approach, we do stereotactic injection of alpha-synuclein PFF, by twin level alpha-synuclein PFF into the striatum, and we take the striatum and do the IP. And we found in both proteomic approaches, we found 256 common protein. And as you can see that in the gene set analysis of 256 protein, both translation pathway and then mTOR pathway along with another catabolic processes, you can see it's significantly enriched in both signaling, in both proteomic approaches. And as you can see in the validation of our proteomics, you can see the immunoplots, some critical component of the mTOR pathway. It shows significantly in both, particularly the TSC, as you can see in the apex the proteomics, as well as in the pull down from mouse brain with alpha-synuclein PFF. It significantly enriched in both proteomics. So as you may know, the mTOR pathway is constantly constitutively downregulated by TSC complex, as you may know in here. So we know in our proteomics that pathologic alpha-synuclein bind TSC2, not TSC1, and we wanted to figure out how these complex actually affected by pathological alpha-synuclein. So we do the collaboration, a couple of labs in Hopkins. So we do the single molecule pull down assay to show the integrity of TSC complex. So this assay basically is the immunoprecipitation on the neutropidine coated glass slide. So we give the protein complexes in presence of alpha-synuclein protein aggregate, both monomer and alpha-synuclein and different protein components. As you can see, and we can see, we can visualize the both TSC and TSC1 and TSC2 in single molecule level, and we can incubate with this IP with alpha-synuclein aggregate, and like say alpha-synuclein PFF. We see that the complex, the TSC complex, is disassembled even in nanomolar alpha-synuclein concentration. This data is really exciting because the TSC complex sits upstream of the mTOR pathway, and then crucial, and it's important in everything: autophagy, protein synthesis, and different other pathways. As you may know, the alpha-synuclein, the mTOR signaling is regulate translation. So we wanted to measure mRNA translation in different model. For example, in vitro reporter assay, if we add recombinant alpha-synuclein PFF, we see increase in translation in concentration dependent manner, as you can see on the top. In the sunset assay in the neuron, in PFF-treated alpha-synuclein neuron, you see huge pyromycin incorporation, significant pyromycin incorporation in the PFF-treated neuron. In the fly brain, we label the fly brain proteins with the radioactivity. It's called S35 pulse assay. So we feed fly with S35 radioactivity, and we wanted to see how much this radioactivity is incorporated into the newly synthesized protein. As you can see, the alpha-synuclein fly, it shows robust incorporation of radioactivity in fly brain tissue, and this is sensitive to the rapamycin. As you can see, it disappeared. And you can see, as I mentioned, this is about mTOR pathway. You can see the deregulation and upregulation of mTOR signaling in A53 transgenic mice in the lab. As you can see, all the substrate we check, the 4E-BP, A6 kinase, mTOR itself is a huge upregulation of phosphorylation of the substrate by mTOR kinase. We took both pharmacological rescue strategy as well as genetic rescue strategy to stop both translation as well as mTOR pathway. In pharmacological approach, we use the anisomycin. It is the general translation inhibitor. It inhibits translation, but it's a very toxic. We use the nanomolar concentration in both neuron and Drosophila. We use A6 kinase to inhibit translation, and we use rapamycin in neuron Drosophila as well as mouse to block mTOR pathway. We use genetic rescue strategy. We overexpress 4E-BP translation inhibitor, and we overexpress TSC in our transgenic fly model in the neuron, so that we can rescue PD pathology. This is one of the piece of data in our mouse model, A53 in alpha-synuclein PFF injected mouse model. So we feed alpha-synuclein injected mice with control feed and rapamycin feed. This feed is commercially available. It's called micro-encapsulated rapa, also called E-rapa. So this food is available commercially. So we feed mice with control feed as well as rapamycin feed. So we start from day one and we follow to up to 6 months. As you can see, the whole brain sections, and you see the alpha-synuclein pathology. We check alpha-synuclein pathology in whole brain section. Left-hand side, the PFF injected with control feed, and right-hand side, as you can see, all the brain section. And the alpha-synuclein pathology phosphorylation of PS129, as you can significantly reduce in all brain section in the PFF mice model. We also examine the neurodegeneration in this mice after 6 months. As you can see, feeding in rapamycin feeded PFF injected mice. As you can see, the rapamycin rescue significantly rescue the midbrain dopamine neuron degeneration. We overexpress, as I mentioned. So this is the genetic rescue experiment. So we take the new generation alpha-synuclein fly. As you can see, this fly is really pathologic. As you can see, the red curve, the decline, the locomotion climbing epicity, even in 15 days, it goes to 50%, as you can see. So this fly, so in this fly model, this model is made from Harvard lab, Dr. Melfini's lab. So we take this fly and we overexpress translation inhibitor 4E-BP and TSC2 in the fly model. As you can see, if we overexpress TSC2 in the Western blot, as you can see, we confirm the expression of 40BP as well as TSC2 in the fly model, as you can see. And then if we overexpress these genes in the fly model, and the locomotion and climbing defects is significantly reduced, and it's become almost normal to the wild-type normal fly, as you can see both in left-hand side in climbing assay with 40BP as well as TSC2. And then the right-hand side, the immunoblots is confirmed the expression of 40BP protein, as you can see, and the TSC protein, as you can see, TSC protein. But these are the human TSC and human 40BP in the fly model. So these rescue strategies work very well in our assay system. So in summary, I want to, so we report a pathogenic state, alpha-synuclein mediated new pathogenic state. So in which alpha-synuclein bind TSC complex and it disassemble TSC2, disassemble TSC complex, and that leads to the persistent activation of mTOR pathway, and then this leads to the 40BP hyperphosphorylation and inactivation of translation repressor, and then it leads to the bulk protein synthesis and proteotoxic effect and the neurodegeneration. And in the rescue strategy, as I mentioned, we take both pharmacological rescue strategy as well as genetic rescue strategy. We overexpress 40BP constitutively, we overexpress 40BP as well as TSC, and it's rescue PD pathology. And in final, in future, we want to study, we don't know what is the genome-wide role of this translation upregulation. So in future, we want to do ribosome foot profile, ribosome footprinting to know what is the effect of genome-wide effect of translation deregulation in alpha-synuclein model. With that, I acknowledge and I thank my mentor and the collaborator and the funding organizations, and finally John Hopkins. And I will stop here, and I will take any question if you have.
That's wonderful, Mohammed. I have a few questions. Why does increased protein translation cause neurotoxicity? Is it because you're increasing the mutant alpha-synuclein translation, or is it some other mechanism?
So we do not know the gene level, what gene is getting more translated, but it's a bulk protein synthesis. So we do not know the whole translatome, which protein is getting more translated. So that's why we want to do the translation. But what we have today is, what we can say is a bulk protein synthesis.
Okay.
Upregulated.
But you're not seeing changes in the level of alpha-synuclein with the rapamycin?
We see a little bit. If we give the rapamycin, we see little less alpha-synuclein protein level, because if we give rapamycin, that's you are inducing the autophagy. So then autophagy effect comes, as well as the translation pathway goes down. So when you give rapamycin, it has multiple effects. It has translation effect. Translation goes down because the 40BP get inactivated, it blocks the translation, and also autophagy is come in this effect in this pathway.
And is alpha-synuclein cap dependent protein translation, which is the one that's inhibited with?
Yeah, so what I have, in the reporter assay, what I have showed, the mRNA reporter we added is a cap dependent mRNA reporter. But in the in vivo neuron and the fly level, we do not know what kind of mRNA is getting translated more or less. So what we say is a bulk protein synthesis.
Okay. And then my last question is, would you predict that TORC1 inhibition would have greater impact on certain forms of Parkinson's disease, like LRRK mutants or GBA mutants or alpha-synuclein mutants? Is there a particular form or any alpha-synuclein?
Yeah, this is a very interesting question. So A53 model also show robust translation in this A53 model, and the genetic model A53, the familial mutation. A53 model. So LRRK2 mutation is already known. LRRK2 mutation is already translation is upregulated, but we do not know the translation-wise or biochemical pathway-wise, the LRRK2 pathway and the alpha-synuclein pathway biochemically interact or not. We do not know yet. We do not know yet, but it is an interesting question we want to pursue in future. We discussed many times whether like the LRRK2 is getting affected by alpha-synuclein, or LRRK2 has an effect on alpha-synuclein to induce translation.
Separate from mTOR.
Yeah.
Excellent.
So I have a couple of questions. Excellent work, congratulations, and for your award. Okay, so the first question is going to be very fast. So do you think, upstream of TSC is AMPK signaling. So do you think by activating like AMPK with metformin would have similar effects?
we didn't follow AMPK. What we saw actually is the THC complex itself is dissociable, and you know the THC complex is a heterodimer. So if this complex is dissociated, this protein start, particularly THC, start getting degraded. And we checked a lot of human brain tissue to see what happened to the THC protein and THC. And THC protein is really, THC2 particularly real, significantly less in all the like dementia brain and the PD brain we checked. But AMPK we haven't seen yet. We haven't seen it.
Okay, and a final question. Would you expect that by upregulating the 4-EBP1 levels, you could get an increased integrated stress response?
That's what already Dr. From Duke University, somebody, so middle of our work. So we know the entire pathway, but so 4-EBP works in every, so in starvation in every way. If you overexpress 4-EBP, so it turn on another survival pathway. So the 4-EBP overexpression, if you bring 4-EBP, is really enhance survival. In PD, it's true for PD because in our model, in neuron and Drosophila, it's true. And that mouse model already, Duke University doctor, so they tested, so in PFF model, they overexpress 4-EBP and in protective. And what I showed in the second slide in the introduction, it's really misfollowed alpha-synuclein stress. It's really get fixed in the alpha-synuclein model, PFF model.
Thank you very much. Congratulations.
Yes.
Thanks.
And now, yes, thank you. And now we move to the final speaker, which is going to be the first award, Jesse Boganic. And Jesse is going to be talking to us today about the biological age is increased by stress and restored upon recovery. So.
Okay, so can you see and hear?
Yeah.
Yes. Okay, perfect. So thank you so much for the opportunity. Metabesity 2020 was like the very first aging conference that I ever attended when I started working in aging. So it's a huge honor to present today. And I'd like to tell you about this study that we've been working on where we try to understand how biological age is impacted by severe physiologic stress. So we know, just based on the last few talks, right, that their biological age is not static, right? So chronic stress, disease, and injury can accelerate biologic age, and there are now, at least in mice, a number of established interventions that can reduce biological age. We wanted to look at this through a slightly different perspective and ask the question, are there situations where you can have a very stressful input that would accelerate biological age, but then after a period of recovery, relax back to the baseline? So I'll just quickly overview the biomarkers we're using to assess this question and infer biological age, and then some of the examples that we've found. So thank you to Max for giving a general overview of the idea of an aging clock. Of course, Max was talking about lipids, and I'm going to talk about DNA methylation. And this, I would say, is a little more developed. Of course, you know, one of the real pioneers was Steve Horvath, and shown here are two of his famous examples, the famous Horvath multi-tissue clock and Grim age. So these are human DNA methylation clocks. They come in a few different flavors. And since the advent of human clocks, there have also been developed mouse DNA methylation clocks. Our lab has contributed some of the first examples using reduced representation by sulfite sequencing. And more recently, there have been microarray platforms developed, which allow us to increase the throughput a bit, which is very useful. And before we jump into the data, I just wanted to talk about this parameter called age acceleration. For those who don't know, so when you do these predictions, if you're right where you expect to be based on the chronological age, you'd call that normal aging. But of course, you can have situations where you're either above or below what you would expect to be at. And it's these deviations that we feel are biologically interesting. And indeed, this is the parameter that we are using to infer biological age. And it's called in the field age acceleration, although it should be noted that it can be either accelerated or decelerated. So we started this study with one of the most established models in the aging space, heterochronic parabiosis. And these experiments were done in collaboration with my colleague Bohan Zhang and Professor Jim White's lab at Duke. And of course, parabiosis is a model where you have two different age mice, one old and one young. You surgically connect them for a period of time. We do a relatively long parabiosis of 3 months. And then somewhat unusually, we separate the animals after the 3 months and allow them to recover for a further 2 months. And as a control, we take 2 age-matched mice, connect them together and subject them to the same conditions. And we started by examining what happens to the livers of the young mice, so either exposed to old circulation or not. And somewhat unsurprisingly, when we apply the methylation clocks to this, we see that when the mice are exposed, the young mice are exposed to old circulation, the methylation age is increased. So that's not particularly surprising, but what was surprising to us was when we allowed them to be separated and recover, we saw that actually very rapidly after just 2 months of recovery, they had reverted to their baseline methylation age. And this is based on a liver clock, so a clock that's trained on liver samples and applicable to liver samples. We also confirmed this with a pan-tissue clock. This is a clock that's trained on multiple different tissue types. And to solidify this result, we also switched from these 2 clocks, which are methylation array-based, to an orthogonal profiling method, RRBS, which is sequencing-based, and we applied an RRBS clock with very, very consistent results across the board. And what's even a little more remarkable is actually based on some very recent data we've gotten, in every tissue we've examined in this experiment, we get extremely consistent results. So from left to right, we have liver, heart, brain, and can't see this because the zoom window is there, but I think that's fat. So it's really a remarkable effect, and it's kind of a proof of concept that you could have, at least in this very kind of artificial experimental condition, a situation where you have a reversible increase in biological age. But another interesting observation that came out of this experiment is that when we looked at the control mice, the isochronic mice, if you compare red to purple, you can see that when we separated these mice and allowed them to recover, we got a smaller but also significant reduction in the methylation age after the recovery. And so this led us to think, could it be not only the exposure to the age circulation, but also perhaps the stress of being subjected to parabiosis for 3 months that led to part of this effect. And so we asked whether this was kind of a general phenomenon in biological age dynamics. And to start asking that question, we turned to a publicly available data set in humans. These are from humans undergoing major surgery. And this first set I'll show you is from elderly individuals who had traumatic hip fractures and required emergency surgery, and they had blood sampled before and after their surgery, and then after a few days just before they were discharged from the hospital. And across a variety of clocks, we got results that are somewhat reminiscent of our parabiosis results. So from pre to post-surgery, we got this significant increase in biological age. This is actually quite remarkable because these 2 time points are separated by less than 24 hours of chronological time. But you can see that by the time they're discharged from the hospital, they're essentially back down to baseline. And also in this data set, we were able to examine patients who had undergone elective colorectal surgery. So the previous set was emergency surgery based on a traumatic injury. These were elective surgeries. The people knew that they were getting surgery. They were selected as good candidates. The procedure was laparoscopic. And you can see that when we applied the clocks here, we got no effect across the board. And so this data set was kind of instructive for us because it made us think that it's the severity of the physiologic stress that's really driving the magnitude of the effect that we observe. And when we asked ourselves what other situations could lead to a similar kind of effect, we were thinking of pregnancy, because of course pregnancy is very physiologically and metabolically demanding. And we started in black 6 mice. We collected blood before pregnancy at 2 time points during their pregnancy, and then after they delivered and had a chance to recover. And we got this very nice trajectory where from before pregnancy to during pregnancy, we get a significant increase. And then after they, when they're postpartum and have a chance to recover, they're back down to the baseline. And we wanted to examine whether this happens in humans as well. And here we had a bit of a technical challenge, which was that we couldn't find a data set that covered the entire period from pre-pregnancy to postpartum. So you'll see 2 panels here. But what we found was that as a function of trimester, we get an increase in biological age. So this covers from pre-pregnancy to during pregnancy. And then in this data set, which covers during pregnancy to postpartum, 6 months postpartum, you can see we get this recovery effect. So I think together, that kind of tells the story. And I'm just showing representative examples here, but actually for pregnancy, we were able to examine quite a number of human data sets, all with consistent results. And finally, we hypothesized, and I'm glad Joan is here, that COVID-19, or more generally, a very severe infectious disease could also cause a similar effect. So we were able to collect samples from 29 patients who came to our hospital, Brigham and Women's, for intensive care during the very early part of the pandemic, March to June 2020. We selected the sickest patients we could find. So these people all came here specifically because they needed intensive care. They were all ventilated and all very ill, yet they survived because we were interested, of course, in the recovery. But we predicted and found that the biological age was increased by the time we were able to collect the first sample. And again, by that time, they were already very ill and in the intensive care unit. So here I'm showing, just as an example, this Dunedin pace clock, which you may know the normal pace of aging in this clock is 1. So you can see that by this first time point, they're already significantly accelerated. But what we found, at least for females, was that by the time they were discharged from the hospital at this point, there was a partial but incomplete recovery for most patients. And in males, we didn't see this effect. What I can tell you across the board is that there was just much more heterogeneity in these trajectories across all the clocks we examined in males versus females. And here's a practical application of what you can do with this data. So we inferred the rate of biological age recovery in this system by comparing the last 2 time points, and we were able to stratify these data by the experimental interventions that these patients received at the time. Just to remind you, this was early days of the pandemic, so hydroxychloroquine and remdesivir were open questions at that point, and tocilizumab was also in clinical trials. And what's interesting is that we did not see effects on the clocks for hydroxychloroquine or remdesivir, but tocilizumab seemed to enhance the rate of biological age recovery during that recovery period. So that may warrant testing as a geroprotective drug. So in summary, I hope I showed you that these biomarkers are very useful for studying kind of what I would call these fundamental facets of the nature of biological age and aging itself. And specifically, we found these reversible changes across these diverse interventions and situations. And I think the broader implication is that biology must have some kind of age reversal mechanisms that allow organisms to recover from these stress-induced increases in age. So in the end, thank you very much to everyone on this slide, particularly Vadim and the lab, our many collaborators who make the work possible, funding. Please check out our preprint if you're interested in more information, and reach out if you have questions. Thank you again for the opportunity to present today, and I'd be happy to take questions.
Fabulous, Jesse. I mean, your work raises so many questions, like what is really biologic age if you can age in a day and then reverse age in a day? What are we really measuring with these biologic clocks? What about when you have a bad day at work and you can sort of feel like you're aging? Like, if we measured a biologic clock, are we older with psychological stress? It's just fascinating. And then when you have companies claiming we have decreased your biologic age by 5 years, was it because the day your baseline you were stressed and then the day you got your next measurement you weren't stressed? It's so malleable.
Yeah,
it's fascinating.
Yeah. You know, so I think that's a huge, if that was a question, that's a huge amount of ground to cover. But I would just echo the last part of what you said, which is that I think that it's a very real kind of issue, I guess, for people who are applying clocks, you know, both, I guess, consumers, but also scientists who are using these to study aging. So, you know, from our perspective, I think we consider it an interesting and, you know, underappreciated facet of the nature of biological age. But, you know, of course, Metabesity is very focused on development and application of biomarkers, right? So if you're a person who's developing or applying biomarkers, it's also something that really needs to be taken into account.
Right. And I mean, there was another wonderful abstract from your group that we heard about yesterday, where half of the things that change with age are good for you. And so you really don't want to reverse that half and decrease that component of biologic age. So there's so much, I think, that your lab is starting to uncover about the complexities of what it means to move these clocks, to reverse these clocks, which components actually we want to reverse, which we don't, and how much they may just, what changes them. It's so interesting.
And what about, okay, so congratulations, Jason. And what about chronic challenges? Because you, apart from pregnancy, all the other challenges that you did were acute challenges.
Yes.
So are you
Yeah,
gonna
so we haven't really studied that per se, but a lot of people have done very elegant work in the field. You know, I think particularly about Dan Belsky and his collaborators. They've studied the effect of chronic stress. So things like chronically living in poor environmental conditions and you know, really severe psychological stress, like serving in the military. You know, it seems to be the case that, you know, just generally chronic stress is bad for your aging, and you see accelerated aging in those cohorts. But you know, what I would like to highlight about our study was that we really tried to focus on longitudinal data sets. A lot of these other studies use cross-sectional designs, which are fine, but of course, you know, you essentially have the chance that you miss these dynamics, right, if you're not following individual trajectories. So, you know, I think that all of those chronic studies, they agree, you know, they kind of fit, but we were interested in the acute stressors because we were interested in kind of tracking these trajectories over time in individuals.
You should do that in your studies too, but be using your clock.
Mm-hmm.
The other thing that your data raises is are these biologic clocks just measuring stress? And as we age, we just don't recover from stress. So it's just a measure of cumulative stress.
Yeah, I mean, it could be the case. You know, one interesting example is that for pregnancy, you know, the other studies have shown that the parity, in other words, the number of pregnancies over the lifetime, that's strongly correlated with the rate of biological aging. So you can imagine a situation, right, where if you have one pregnancy, you have the stress, and then you have an incomplete recovery, then you have the next pregnancy, on and on, you can kind of offset the rate of aging, if you will. So yeah, I think it's an interesting question. We don't have direct kind of data to answer that at this point, but it's certainly a sort of natural direction based on the questions that arise because of this work.
And I just thought, did you look, how old were the people having the acute trauma surgery where you saw that?
Yeah, so very, very interestingly, that data set was all elderly people. So the average age was around 75. Wow. And we tried to stratify kind of many different ways, but you know, one of the ways was we tried to stratify the recovery rate by age, because we kind of expected, you know, that maybe if resilience is declining as a function of age, you'd have a less efficient recovery. We didn't see that, but I'm not really confident including anything at this point because it was a relatively small cohort of people. But again, I think it's a natural question to pursue further. So
David, do you want to ask some questions?
Yes, I do. Great presentation. I have there's hope, right? That's a good. So recovery appears to have a major effect on deceleration of aging, at least on those clocks that you looked into. I have a question that's a very technical question. What has been the largest change that you've seen with this chronic or psychological stressors, or maybe even surgery? Would you say it's 50% of their age? Would you say it's twofold, 40%? I just want to have a kind of a rough idea on how much these things fluctuate upon stressors.
Yeah, so it's a good question. So parabiosis, I would say, is the largest effect, but I don't know if you would want to necessarily chalk that all up to stress, right? Because you've also got the systemic factors from the old circulation doing whatever it is they're doing. For the human data sets, which is the largest effect, I would say it's a tie between pregnancy and the major surgery, at least the trauma surgery. And, on average, the effects there are kind of between 2 and 3-fold from kind of the baseline to the high points.
That's why I think we are inventors of the inflammatory age clock, and we've seen a lot of different changes in different circumstances. Sometimes 50% of the inflammatory age years, and I always wonder, to what extent that's fair to say that this is a biological age clock.
Right.
If we so. Back to John's question, and I echo that. These things are so easy to change, right? And so the other question that goes hand in hand with that claim is: have you ever looked into cell proportion changes? Because in our hands, some of these methylation clocks are largely dependent on cell proportions and differentiation. So say you have the effect of COVID, and then you have a massive increase in particular cell types, and then all of a sudden, because those CpG islands are associated with differentiation, you have a huge increase in your biological age, but that's just a rather a result of changing in cell proportions.
Yeah.
So have you looked into correcting for that?
Yes. So, this is an excellent question because, of course, there are two possibilities, right? One is that, two broad possibilities. One is that you're affecting biological age. One is that you're gaming the biomarker, right? So yes, we have looked into that. All of the human data, of course, is based on blood, and of course, blood cell composition. As you say, it's a very real possibility. So two things that I can say about this. Number one is that based on the methylation data, we use these methylation predictors of blood cell composition, and we applied those across the board. And essentially what we found was that we see these kind of very consistent effects across multiple different clocks, across the different models. Those are very consistent. But when we look at the changes in blood cell composition, at least based on the methylation predictions, we did not see consistent effects across the different models. And so we don't really believe that that cell composition effects alone are sufficient to explain the data that we have. The other thing I would say is that the clocks are capturing blood cell composition, at least in part. This is I don't know if I would necessarily say it's by design, but it's known. And I'm not sure that I entirely kind of disagree with the idea that that itself is not a manifestation of biological aging, right? Because it's part of age-related changes, right? That you have these blood cell compositions. So that veers a little into the philosophical, I guess, but it's a very relevant concern, and we try very carefully to control for those things as much as we can.
Yeah. Okay, that's fair. Thank you, sir. Thank you, Jason.
And what I also wanted to comment is the fact that it was very interesting, even in the few patients that you examined, the COVID patients. I like the fact that it goes very well with what data David showed us earlier on today about the inflammation clock. So when you actually block just one cytokine, isn't it, IL-1 beta? So you see the reversal, whereas you don't see a similar effect with hydroxychloroquine or with the other treatment, which is interesting.
Do we announce the winners, or do
we announce the winners and we announce the sponsor? So you start first.
Oh, no, this is you, Georgina.
Well, it's David. Actually, first of all, I would like to thank our scientific committee, David Furman and Ana Maria Cuervo, for choosing amongst very good extracts that we have received. So first of all, our judges, I want to thank them a lot. Second, I would like to thank our sponsor. So Kinexum and Zan Fleming are the sponsors of these awards. And now you can announce the winners. No.
I am taking Ana Maria Cuervo's place, who couldn't, unfortunately.
So David, you announce the winners.
Yes.
Oh, do I have to do that?
You do.
Well, I would have to pull that from
Okay, I'm gonna help you. So
Yeah, help me, help me.
okay, so starting from the third place is Maximilian Anfried. So congratulations, Maximilian, for getting the award of shall I say the amount? $1,500
Wow. And
Thank you.
Maximilian, this is out of a lot of
Yeah,
different.
Maximilian, come into
You're
the
the top
camera.
3. I'm jealous.
Yes, I know. Congratulations.
Congratulations.
Done.
Thank you.
Okay, the second.
Second place is Mohammed Khan.
Yes, please turn on your cameras.
Mahmoud, who's running a $20 billion fund.
So, that's really good.
So congratulations, Mohammed.
Thank you so much again. I cannot turn on my video. I don't know what happened. It said you cannot start. the
Okay, no
video
worries, but
because-
at least we wanted you to try. Thank you.
Okay,
We
thank you.
get
so
to
much.
announce his.
So Mohammed got the 2,000.
ma'am.
Congratulations, $2,000. Congratulations for your work.
Thank you.
And
Thank you so much.
And first prize is Jesse. Fabulous work.
Congratulations,
Thank
Jesse.
Thank
Congratulations,
you.
everyone.
And Jesse got $2,500.
Go.
Excellent work.
Have fun.
Thank you. To treat my lab mates to lunch, at least.
Okay.
Nice
Even
lunch.
better. Wonderful. Thank you, everybody. Such good science being done by you. You're the next generation of aging stars. We're like so excited to watch you guys continue to fly.